First off, a helicase comes to the DNA and splits open the hydrogen bonds between the individual nucleotides. Next, an RNA polymerase comes and binds the promoter and adds nucleotides based on the base pair rule. It stops at the terminator and leaves. Then, another RNA polymerase comes back and checks the new RNA. Now that it's checked for mistakes, it's ready to go. The process of protein synthesis continues with the next step of RNA processing. The process also occurs in the nucleus. First, the protective cap and poly A tail approach the wild mRNA strand. The cap and tail provide protection against deadly enzymes. We can see here that the mRNA strand is made of exons and useless introns. SNPs assist in removing these introns from the mRNA strand because they do not code for our end product, a protein. Next, the SNPs help slice together the exons and the chain is formed. Up next is translation. First, mRNA slides into the frame. This mRNA grabs a small ribosomal subunit. Next, a tRNA, transfer RNA, attaches to the start codon. After this, a large ribosomal subunit attaches to the P site. The ribosomal unit then reads the mRNA. A triplet mRNA base, a codon, is positioned on the P site. The first codon is a start codon. A complementary tRNA carries an amino acid and attaches its anticodon to the mRNA codon of bases. Then the entire ribosome moves to the next codon until a release factor comes and releases it. Next is protein processing. The now fully grown polypeptide chain makes its way over to the Golgi body. First off, the amino acid methionine is removed from the chain along with any other unnecessary groups. This will activate the polypeptide chain. Next, the Golgi body uses hydrogen ionic and covalent bonds to fold the polypeptide chain. The primary stage is a straight chain of amino acids. Secondary, the chain starts to fold. In the tertiary stage, the entire chain folds into its 3D shape, and now we have a protein.